Okay, let's jump right in. Welcome to the deep dive. Today we are exploring, well, a really foundational piece of research, I think, in environmental monitoring. We're plunging into the paper. Environmental DNA, eDNA removal rates in streams differ by particle size under varying substrate and light conditions. It's led by Elise D. Snyder and her colleagues. Our mission today is basically to get our heads around the physics and biology of this genetic material floating around in water. We're talking eDNA, shed skin, waste, tiny tissue fragments that critters leave behind. We need to figure out how quickly that stuff gets, removed from flowing water. And how things like what's on the stream bottom or how much sunlight there is affects things based on how big the actual DNA particles are.
Yeah, and the context here is really fascinating. eDNA is, it's already changing the game. It's pretty cost effective. You don't have to disturb the animals. Great for tracking invasive species or finding really rare things without ever needing to, physically catch them. But the challenge comes when you move from still water, like a pond, to moving water, a stream or river, what we call a lotic system. That unidirectional flow, it just makes things tricky.
How so?
Well, say you take a water sample way downstream. You get a positive hit for, some specific fish DNA, pinpointing exactly where that fish is or how many there are. That's still a major headache. The water just carries the signal away, you see, and we really need to understand how quickly that signal fades or sinks or breaks down before we can reliably use eDNA to map where things actually live in a river.
Right, so that's the core problem. If the water is flowing steadily, what makes the DNA signal weaker downstream? The paper calls it removal, right? But is that just the DNA breaking down chemically? Or is it literally sinking?
It's actually both. What scientists mean by removal here is this complex mix. Part of it is physical, the DNA particles getting stuck on rocks, sinking into the mud, that kind of thing, deposition. And the other part is biological decay, microbes munching on it, enzymes breaking it down. And because how much of each happens really depends on the specific stream, the amount of eDNA you measure downstream doesn't always neatly match up with how many animals are upstream. It's inconsistent in flowing water.
Okay, that leads us to something really key in the paper.
Yeah.
eDNA isn't just one thing. It's not uniform. When an animal sheds DNA, it's more like a collection of different bits and pieces.
Exactly. It's what they call polydispersed particles. Think about, dropping a biscuit in the water. You get fine crumbs, some smaller than zero point 0.2 micrometers, but also bigger chunks, maybe over 180 micrometers. These come from slowed skin or feces, mucus, all sorts of sources.
Different sizes, okay.
Yeah, and this brings up a really critical question. Does the size of the particle change how far it travels? Basic physics tells us a particle's properties, its size, its density, affect how long it stays floating versus how quickly it settles out. So if we figure out the different particle sizes in a sample, maybe that tells us something about how far away the source is.
Ah, okay. So to test this, the researchers needed to, control everything else, right? They wanted to isolate how light and what's on the stream bottom, specifically these things called biofilms, affect the removal of different eDNA particle sizes.
Precisely. And their setup was pretty ingenious. They basically built these mini streams, 24 of them actually, little recirculating fiberglass channels, they're called mesocosms, set them up at the University of Notre Dame. They then added a known amount of eDNA from common carp into these controlled streams.
Okay, mini streams. And they manipulated conditions.
Yeah, a fully crossed design, meaning they tested every combination. 6 unique treatments in total, with replicates for each. They changed 2 main things. First, light. Either shaded blocking about 90% of the photosynthetically active light or open, so getting natural sunlight, although the greenhouse glass did block most of the really harsh UV rays.
Okay, light or shade. What else?
The substrate, what's on the bottom? They had 3 types. 1, just water, no substrate at all. 2, bare substrate, basically fresh, clean gravel and cobbles. And 3, biofilm colonized substrate.
Biofilm, like the slimy stuff on river rocks.
Exactly. They let rocks sit in stream water for 6 months beforehand, so they developed this nice, thick, natural layer of microbes and algae, really mimicking a mature stream bottom.
I get the need for control in the lab, setting up these pristine mini streams. But, how much does an experiment in a fiberglass tub really tell us about a messy, real river? With all the swirling currents, muds, temperature changes, things they removed.
That's always the trade-off with mesocosms, absolutely. You gain control, you lose some real-world complexity. But the key here is isolating specific factors. And to make the data meaningful, their collection method was crucial. This is where the sequential filtration comes in. It's pretty neat. They took water samples at different times, 40 minutes, 6 hours, 18 hours, and 48 hours. Then they passed each sample through a series of filters, one after another. First, a 10 micrometer filter, then 1.00 micrometer, and finally a very fine 0.2 micrometer filter.
Ah, so sorting the particles by size.
Precisely. This allowed them to measure how much CART eDNA was in each size category, large, medium, and small, at each time point. So instead of just saying total eDNA is decreasing, they could say, right, the big particles vanished really quickly in this condition, but the tiny ones stuck around for ages. That level of detail is what starts to bridge the gap to real rivers.
Okay, let's get to the results then.
Yeah.
What about the total eDNA removal, like lumping all the sizes together?
This is where it gets really interesting. The single biggest factor, hands down, was the biofilm.
The slimy rocks.
Yep, the biologically active substrate. When biofilms were present, the eDNA just disappeared incredibly fast.
How fast are we talking?
Well, they calculated a removal rate constant, K. With the biofilm substrate, K was about 10.2 per day. Compare that to the bare rocks, K was only 2.0 per day. And with just water, no substrate, it was even lower, 1.1 per day.
Wow, so nearly 10 times faster removal with biofilm compared to just water.
Exactly. And think about what that means in time. The average eDNA particle, when biofilm was present, was basically gone from the water column in about 2 hours.
2 hours.
Yeah. But in the control tanks, with just water, it took around 22 hours for the same amount of removal. Huge difference.
So what are the biofilms actually doing?
They're like super efficient eDNA traps. Two main things are happening. First, physical trapping. Biofilms are sticky. They have this matrix of polysaccharides, like a net that just grabs onto particles flowing past. Second, biological breakdown. Biofilms are packed with microbes, bacteria, algae, fungi, and many of these microbes can actually use DNA as a food source. They release enzymes like DNases that chop up the DNA.
So they trap it and eat it. A double whammy.
Pretty much. And it's interesting, even the bare rocks showed faster removal than just water alone. That tells us that even simple physical settling the DNA, just bumping into surfaces and sticking or settling in crevices, is a significant removal process in flowing water, even without the active biological component.
Okay, and what about light? Did that matter?
It was a bit more subtle. Overall, statistically, it wasn't the main driver like substrate was, but there was an interaction. When biofilm was present, having light did speed things up. In the open tanks with biofilm, the eDNA signal dropped below detectable levels faster, usually within 18 hours. In the shaded tanks with biofilm, you could still detect it after 48 hours sometimes.
And that's not UV breakdown because the glass blocked most of that.
Right. The thinking is that the light was probably boosting the activity of the algae and other photosynthetic organisms within the biofilm. More photosynthesis, maybe a more active, stickier biofilm, or perhaps the microbes consuming the DNA were just more active in the light. It suggests light enhances the biofilm's cleaning power.
Okay, so substrate, especially biofilm, is king for total removal. But you mentioned the real novelty was looking at particle size, right? Especially in flowing water.
Yes, this is the part that really contrasts with some previous work. Remember how we said eDNA comes in different sizes?
Yeah, the crumbs and the chunks.
So in the tanks with just the bare rocks where physical settling was important, but biological breakdown was less intense than with biofilms, they found something striking. The larger particles, the ones bigger than 10 micrometers, were removed much, much faster. Their removal rate K was about 15.7 per day.
Faster for the big ones.
Much faster. Compare that to the smallest particles, the 0.2 micrometer ones. Their K was only 2.9 per day, and the medium 1.0 micrometer ones were 3.9 per day. So the big stuff disappeared way quicker than the small stuff.
Huh. That feels backward somehow. I thought smaller things might break down faster.
That's what some studies in still water, like ponds or lab beakers, had suggested. Maybe because smaller particles have more surface area relative to their volume for enzymes to attack. But this experiment was in flowing, recirculating water, and that changes the game. What this suggests is that in a flowing system, the physical process of settling deposition is often more dominant than biological decay, at least initially. Bigger, denser particles simply sink out of the water column faster. They hit the bottom before they have much chance to decay biologically. The tiny particles, though, they stay suspended in the flow for much longer.
So gravity and flow dynamics overpower the decay rate, especially for the big bits.
It certainly seems that way based on these results. Physical deposition rules for the larger fractions in moving water.
Okay, but wasn't there a weird wrinkle? Something about the DNA concentration actually increasing sometimes. The breakout phase.
Ah, yes, good point. They did observe that in some tanks, particularly the ones with no substrate, the total eDNA concentration measured seemed to go up briefly around the 6-hour mark before it started declining properly.
That sounds like contamination or bad mixing.
They checked for that. The mixing was good. It's more likely what we call eDNA transformation. Imagine you start with a fairly large but maybe loosely bound chunk of eDNA, like from mucus or something. Initially, it might be too big or structured to even be captured efficiently by their sampling or pass the first filter, but as it tumbles around in the water for a few hours, it starts to break apart into smaller, more easily suspended fragments.
So it's not more DNA, it's just becoming detectable, breaking into sizes the filters can catch.
Exactly. It shifts from being in a large, less detectable form to smaller, filterable, and thus measurable particles, so the measured concentration spikes temporarily. It's a good reminder that eDNA isn't static, it's physically changing in the water column too.
Right. So pulling this all together, what does this super detailed look at size, settling, and biofilms actually mean for someone out there trying to use eDNA in a real stream?
This is where the potential is really exciting. The study strongly suggests that the particle size distribution, the PSD, the mix of big, medium, and small particles in your sample, could become a powerful tool, a tool for figuring out where the DNA came from.
Like a forensic tool for locating the source. How?
Well, think about the transport. They used their measured removal rates, how fast each size class disappears, to model how far downstream each size fraction might travel. Their calculations suggested that the small particles, the 0.2 to 1.0 micrometer ones, could travel maybe 2 to 9 times farther downstream than the big particles, the ones over 10 micrometers.
Okay, 9 times farther.
Yeah. So imagine you take a water sample, you measure the eDNA, but you also measure the proportion of large versus small particles. If your sample has a relatively high proportion of those large, fast-settling particles, it's a strong clue that the source organism is probably quite close by, because those large particles just don't travel very far before they sink.
Ah, I see. But if the sample is mostly made up of the tiny, slow-settling particles?
Then the source could be much farther upstream. This could help distinguish between, say, a small population living right nearby versus a larger population much farther away. Both scenarios might give you the same total low eDNA concentration, but the particle size breakdown would tell a different story about location. It adds a spatial dimension.
That's a potential game changer, moving beyond just presence-absence. But let's be fair, they did mention limitations, right? Their travel distance models were based on the flow speed in their lab tanks.
Absolutely, and they acknowledge that. Their lab mesocosm flow, around 0.19 meters, is probably smoother, less turbulent than many natural streams. In a real river, turbulence might keep particles suspended longer, meaning they travel further than the simple lab model predicts.
So the exact distances might change in the wild.
For sure, but the crucial point is the pattern, the relative difference. Large particles will settle faster than small particles. That fundamental physics holds. The next step, obviously, is to take this into the field. Measure removal rates and PSDs in actual rivers with different flow conditions, different species, different substrates, and refine these models. But this study provides the critical proof of concept and the methodology.
Okay, so wrapping up, the big takeaway seems to be that the fate of eDNA in streams is this really complex interplay between the stream environment, especially the substrate and its biofilm community, and the physical nature of the DNA particles themselves, particularly their size.
Definitely. And the power of active biofilms as these natural water purifiers for DNA is incredibly clear. They remove it so much faster than physical settling alone.
So for you, the listener, this deep dive really highlights that just detecting if a species' DNA is there is just the beginning. The cutting edge is moving towards understanding the state of that DNA using particle size almost like a biological mile marker in the water.
Yeah, exactly. It's about adding that spatial resolution. Flowing water makes eDNA work harder, but by understanding these particle dynamics, treating DNA not just chemically but physically, we unlock much more predictive power.
Which leads to a final thought to chew on. We saw how important biofilms are. What happens in a real river during, say, a massive flood, when huge amounts of sediment scour the riverbed or turbulence rips those biofilms away? Could that cause a sudden massive release or prevent the removal of eDNA, creating these temporary bursts or lags in the signal that could totally throw off our nice, neat, predictive models? And how will future technology, maybe sensors deployed right in the river, cope with that kind of real-world dynamic signal to give us reliable location info on the fly?
That's the frontier, isn't it? Integrating these lab findings with the full complexity and frankly, the messiness of natural systems. It's a huge challenge, but the potential payoff for monitoring aquatic life is immense.